All right, now what we're going to look at is hybrids and the environment. Basically, when we talk about hybrids, we're talking about a species that is kind of a combination of two closely related species. And when that happens, sometimes you can create a condition called hybrid vigor. Hybrid vigor happens when an offspring from two closely related species are actually more viable than the parents are for survival, where maybe they're bigger, maybe they're stronger, maybe they produce better fruit, maybe they produce more seeds. Something that gives them a slight advantage for either us as humans or for them as a plant in order to survive. And in the agriculture world, this is really big. This was kind of discovered by Charles Darwin. When he looked at two different varieties of corn, he bred those two varieties, and the hybrid corn, which if you think about a hybrid is kind of a combination of two things. When that hybrid corn was actually bigger and produced larger, sweeter kernels than either of the two parental generations. So this is something that tells us that phenotypes aren't always determined by just a simple single set of genes. A lot of times there's multiple genes interacting that create these complex phenotypes that result in something called hybrid vigor. And this is where genetics gets a little bit more complicated. That's not just as simple as Mendel said, dominant, recessive, you're either tall or you're short, you're round or you're wrinkled. And when we look at that, we look at something called a quantitative trait loci. We remember that loci is a location on a chromosome where we see a phenotype being expressed from a specific gene. Quantitative means we're going to be measuring something. So we're going to be measuring the locations and the traits based on what they produce. We're going to be looking at the complex character that's being produced by these things. And when we use quantitative trait loci, we're going to be measuring something. So like in agriculture, we compare how much grain is happening in a field as opposed to just counting seed shape and looking at very specific things, because again, when we're looking at this, it's more has to do with how the different genotypes interact to create a complex phenotype, which I know that one gets a little bit confusing, so just focus on the word quantitative. Anytime you see that word, think of numbers, think of measuring, and that's what we're doing with quantitative trait loci. We're going to be measuring the outcomes as opposed to just looking at them and seeing, is it green, is it yellow, is it wrinkled, is it round, looking at simple things. This is stuff that's caused by multiple genes interacting to create these hybrids that have different outcomes from their parents. Now, when we look at the environment plays a huge role in what genes are actually expressed, and this has to do with complex phenotypes. Again, Mendel did a great job getting us to where we are, but he was far, far from perfect. Not very many genes are just simple dominant recessive. You may have a dominant trait that doesn't show up because it's tied up on a histone. You may have a dominant trait that doesn't show up because you don't put it in the right conditions to be to express itself. I mean, things like height. If you don't feed your kid enough food, they're only going to get so tall. They have to have as much nutrition as possible to fully express that gene. And that's where the environment can play a big role. If you're in a war-torn country, you're going to live in a place where your body's stressed and your body has to focus on defending itself rather than producing things. And when we get to later on chapters about evolution and how things sort of survive, we'll talk about the allocation of resources and how that's important for an organism and has to do with the things going on around them. Um, Another thing that we can measure in the environment is something called penetrance. Penetrance tells us how often a gene shows up in is expressed in someone that actually has it. Excuse me. How often the gene's expressed in someone that has it. The common example that your book uses and something that's out there a lot is a BRCA1 gene or the BRCA1. This is present in women that have breast cancer a lot of times, or a mutant allele of it is present in women that have breast cancer, I should say. But here's the thing, not everyone that has that gene is actually going to test positive for breast cancer or express the breast cancer gene in their lifetime. A lot of it depends on environment and what actually goes on. So our penetrance is measuring how many people have that BRCA1 and how many people actually show it, and we get sort of a ratio there. Expressivity tells us how the extent to which it's actually put out in the phenotype. Do you just get breast cancer, or do you get breast cancer in combination with other things? What is the extent to which that gene actually shows itself? And that's what expressivity is showing us. It's how severe the symptoms are associated with the gene. Maybe not severe, just how magnified is the gene inside your phenotype. And that's all we're going to do with these ones, and we got a couple more presentations on this chapter later.